Good afternoon, ladies and gentlemen. Bon après-midi, Professeur Petit. Welcome to Sky Lounge Prince in the heart of Antwerp for the conferral of one of the honorary degrees for 2021. My name is Anke. I'll be your hostess this afternoon in this beautiful location. As you may know, this honorary degree should have been awarded 1 year ago on the 2nd of April 2020. But the pandemic put an end to that plan. We are delighted to be able to confer the award this afternoon in this way instead. Honorary Dr. Christine Petit will shortly be teaching a masterclass titled Hearing from the Genetic Dissection of Molecular Mechanisms to a Cure. Afterwards, we will hold a Q&A session to answer the questions you send in through the registration module. If you have any questions during the masterclass, you can also ask them using the chat function on the YouTube channel. Then we will proceed to the conferral of the honorary degree. But first, I would like to give the floor to Professor Koen Augustijns, Dean of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences.
Thank you. Dear Professor Petit, dear Professor Avant, dear Rector, dear Professor Van Kamp, dear participants online, as Dean of the Faculty, it's a real honor to introduce this ceremony. It's really a big honor, and we already thank you for accepting and acknowledge you in advance. Indeed, as was mentioned, we thought of having this ceremony physically. When we postponed it a year ago, we thought, well, let's have the plan to do it a year later and then do it physically. But yet here we are, and it was mentioned, it's a fantastic view. It's a pity that you cannot share with us the view over Antwerp, 13 floors above Antwerp. It's really fantastic. But I'm sure there will be screenshots later on so that you can experience a little bit of Antwerp. So here we are in an online setting. The virus was too smart, and the virus is very tiny. It has only a few genes. So we can only imagine how complex it must have been for you, Professor Petit, to decipher the genome of the hearing system. It must have been a complex task. It's really an achievement that you did that. It's really an honorable task, so we thank you for that. It's a big achievement, and I'm pretty sure that during the masterclass, our students, our young scientists, can learn a lot from you. So this is the opportunity for me to introduce Professor Guy Van Kamp, the promoter of this honorary doctorate, and he will introduce you to the masterclass and give the laudatio for the candidate. So the floor is to Professor Van Kamp. Thank you.
Dear Professor Petit, dear Rector, dear colleagues, friends and students, it is a great honor and pleasure for me today to present the laudatio of this ceremony. Such a laudatio is actually an explanation exactly why the achievements of the candidates are so special, that we believe here at the University of Antwerp that this candidate deserves a very special honor, an honorary doctorate. In this case, it's not a difficult task, as the scientific achievements of our candidate, Professor Christine Petit, are really outstanding. Professor Christine Petit is a geneticist and a neuroscientist. She's a professor at Collège de France and professor classe exceptionnelle at the Pasteur Institute in Paris. She currently directs the Genetics and Physiology of Hearing Laboratory at the Institut Pasteur, and she was the reason and the driving force behind the foundation of the Hearing Institute, Institut de l'Audition, a brand new research center that opened recently in Paris, of which she is the director. So what has Professor Christine Petit and her laboratory actually achieved that is so special? Over the last 20 years, Scientists all over the world have unraveled how our ear works, how we hear, up to the molecular level. Today, we know which genes are involved, which are the proteins, the actual building blocks in our ears that make us hear. We also know how these proteins work and interact, and in addition, we know what can go wrong and what can cause hearing loss and deafness if something goes wrong. It turns out that the human ear is very complex, and it has been a very difficult puzzle, taking many years to solve. One of the most important researchers in this area, someone who has put together a very large number of the pieces of the puzzle, is Petit. And there is no doubt that she is a leader worldwide in this field. I will not list all the important findings from her laboratory because it would be too many. But let me give you an example from my own experience. At one time, about 15 years ago, I was contacted by Christine, and she had found a gene responsible for deafness in the family, and the gene was called PVAKIN. And because this gene was related to another gene that was found and studied in Antwerp, we performed collaborative research on this gene between our laboratories, and this resulted in an important publication describing this new PVAKIN gene. However, Christine did not stop after the identification of this gene, and she went on studying this gene, trying to find out exactly why it was causing deafness. And she did this by making a mouse model for this gene. And then she discovered something amazing. When breeding the PVAKIN mice, she found that the size of the nest, the number of pups in the nest, was affecting the hearing of the mice. Little mice that had many brothers and sisters were hearing very poorly, while mice that were alone or had only one brother or sister, they were hearing much better. And this was very strange, and she investigated further. And she found that the pups, the little mice, were actually normal hearing when they were born, but they had very fragile ears, and these ears were quickly damaged by noise, even normal noise, not too loud, that normally should not damage an ear. And she found that the peeping noise that the little pups made in the litter, only when they were just a couple of days old, was damaging the ears and was making the pups deaf. And the more pups in the litter, then there was more noise, and there was more noise damage to the ears. This is really remarkable, isn't it? And she then studied the ears of these mice with very sophisticated techniques so that she discovered how the PVAKIN gene worked and what exactly went wrong in the ears of these mice. And through her research, she could explain the whole process up to the level of the proteins and biological processes that were involved. I remember a scientific conference of ENT doctors and audiologists where Christine presented these results. And after the presentation, the audience was amazed, and they were highly enthusiastic about the scientific value and the detail of this finding. And people couldn't stop talking about it. And doctors were speculating and thinking of the consequences of this research for their patients suffering from hearing loss, often patients with noise damage to their ears, and whether this new knowledge could perhaps be used to develop new treatments. This PVAKIN story is, of course, not the only breakthrough that was realized in the laboratory of Christine Petit. She discovered many other deafness genes and investigated how they work in the ear. And by doing this, she has been paving the way for new therapeutic approaches. And over the last few years, she has been developing approaches for gene therapy to treat hereditary deafness by gene replacement, providing proof of concept for this treatment. It is a great opportunity to have Professor Petit with us today for a masterclass. And I propose that now we listen to her to hear more details of her intriguing research. And also I would like to remind the audience that questions can be suggested for after the presentation through the chat. Professor Petit, you have the chair now.
So, many thanks to Professor Kuhn Augustin, Dean of the University of Pharmaceutical, Biomedical, and Veterinary Science, and Guy Van Kang, many thanks for your very kind words of introduction, and you're welcome. I am delighted to have this opportunity to share with you our progress in the understanding of hearing mechanism and hearing disorder, and to show how these findings are paving the way for a new challenge, curing hearing impairment. What is hearing? A mechanical sense that detects any element vibrating at an audible frequency. It localizes it, it identifies it. If these sounds are emitted by prey or predator or potential sexual partners, the hearing contributes to the survival of the individuals and species, as the other sounds do. However, during the course of human evolution, hearing has acquired a unique status among other senses. This was the result of the emerging of language grounded in an unparalleled ability to make sense out of sound, which itself involves the development of cognitive function. We spend much of our life communicating orally, at least 20%, even more for some of us. Humans have developed a second sound-based mode of communication, music, which can convey emotions. Thanks to language and music, hearing becomes the primary sense for intellectual and emotional communication between humans. Both language and music play essential roles in building and maintaining social links, thus conferring an additional essential role on hearing. Here is an oversimplified representation of human afferent auditory system. All sounds induce eardrum vibration, which are transferred to the auditory sensory organ, the cochlea, this snapshot organ. From the cochlea, the electrically encoded sound information flow through the afferent auditory neuron here in blue, through the various nuclei of the auditory sensory system up to the auditory cortex. In humans, the encoded sound information is delivered to the primary auditory cortex here in the HLA gyrus in less than 25 milliseconds after sound exposure. The auditory pathway is much more complex than shown here, especially, it contains at least 14 major central nuclei and neuronal fiber of the auditory system are crossing the midline at almost every level in order to be able to compare the auditory information collected by each ear. The physical parameter of sound, frequency, intensity, and phase are extracted in the cochlea. Additional information is extracted beyond the cochlea, and these various components of information contribute to the formation of a percept, an auditory percept, that is itself modulated by other components such as attention, memory, and multimodal sensoriality. The basic principle of the organization of the auditory system is the spatial representation of the frequency, let's say the topographic representation of the frequency, which are also called tonotopic map, shown here at the level of the cochlea and at the level of the primary auditory cortex, and also present all along the auditory pathway. The major physiological characteristics of the auditory systems are the extreme speed with which is processed sound, its extreme temporal precision and reliability, and also its extreme sensitivity. The sensory system has many exceptional performances, but unfortunately, it is also highly vulnerable. Severe deafness has major consequences regarding of the age at which it occurs. Children profoundly deaf at birth cannot spontaneously acquire language and they will face major difficulty in learning acquisition at school. Young adults who become deaf lose social links with their surrounding, and let's say they can develop some depression, and these individuals encounter major difficulty integrating in the workplace. Adults who lose hearing with age also encounter social isolation, and they can especially become dependent. The occurrence of hearing loss in the middle age is today very well established as the major risk factor for the development of cognitive decline and dementia. A huge number of people are affected by hearing impairment, according to WHO. 460 million people suffer from disabling hearing impairment, and this ranks the hearing defect as the most frequent sensory defect. More than 1 billion young people between 12 and 35 years of age are at risk of developing hearing loss due to sound overexposure, mostly in recreational setting. By 2050, 900 million people will be affected by hearing disorder, mostly presbycusis, that is age-related hearing loss, due to the aging of the population and also overexposure to noise, as it has already briefly introduced by Guy Van Camp, especially due to the noise pollution in overcrowded cities, especially in developing countries. In the early 1990s, much was already known about the biophysics and the physiology of the cochlea and its afferent innervation, whereas the molecular mechanism underlying sound processing in this structure remained virtually unknown. The reason was the very small number of each cell in the cochlea, which preclude the use of biochemical or even classical molecular genetic approaches to decipher this mechanism. For instance, a mouse cochlea has a human cochlea contain only about 3,000 genuine sensory cells. Fortunately, this is exactly the type of difficulties that genetics can overcome. The efficiency of the genetic approach is indeed totally independent of the number of cells and molecules being involved in the process study. We chose to search for causal deafness genes in humans in the hope of gathering information of direct information to the benefit of the patient. At the time, no gene responsible for profound isolated deafness, also referred as non-syndromic deafness, had ever been mapped to human chromosome, and this was the first step towards the identification of causal disease gene. This was mainly due to the fact that in Western countries, deaf people, mainly because they attend the same school, tend to intermarry. And it's such a family, the families they form, the tracing of the transmission of a defective gene inherited from the father or the mother could not be achieved because standard clinical features such as the measure of the hearing threshold cannot differentiate reliably between different genetic forms. Given our interest in auditory neuroscience, we focus on profound form of deafness, which can only be of sensory neural origin. They cannot result from external or middle ear defect. There was evidence that profound hereditary deafness forms are mostly transmitted in an autosomal recessive form and are therefore highly prevalent in consanguineous family. To minimize the likelihood of having to deal with several causal genes segregating in the family, we focus on consanguineous family living in geographic isolation, as such communities have generally been founded only by a small number of individuals. Here at the bottom, we can see the pedigree of the first family we studied, from which we map the first gene responsible for deafness to human chromosome. This way, we were here, let's say, in the South Tunisia. Human genome sequencing were very rudimentary. The sequence known was very rudimentary at that time. We thus decided, let's say, to proceed by a candidate gene approach, focusing on genes which are preferentially or specifically expressed in the cochlea, thinking that likely these genes play a critical role in the cochlea and when defective should lead to deafness. We obtained several genes restricted with an expression restricted or preferentially or more important in the cochlea that we match to our human deafness loci, and so doing, we were rapidly able to pull out a number of causal genes responsible for deafness. Today, thanks to the effort of numerous laboratories worldwide, close to 140 genes causing monogenic forms of isolated hearing impairment in humans have been identified. An even greater number of genes responsible for syndromic deafness, in which deafness is associated with other symptoms, have also been found. We were able to conclude that most congenital or prelingual forms of profound deafness in Western countries are of genetic origin. Most of the proteins encoded by these genes were unknown. How to decipher the role of this encoded protein? Not in humans. The human cochlea cannot be directly observed, and clinical auditory tests provide only rudimentary information. We therefore needed animal models, genetically engineered animals, mostly mice, carrying mutation in the orthologs of the human deafness genes. Fortunately, with few exceptions, this mouse model faithfully reproduces human hearing defect. Making a full use of these models requires multidisciplinary approaches. Piecing together the puzzle, we attempted to investigate the complex form by the protein encoded by this deafness gene as probably more informative than individual protein for developing functional hypotheses. To this end, we initially focused on a syndromic form of deafness. In a syndrome, the presence of associated defect further defines the role of the causal gene, and the different causal genes for a syndromic form of deafness are likely to be involved in the same function. We focus on Usher syndrome. Usher syndrome is a multisensory disorder with 3 clinical types. Type 1 is the most severe, and let's say in type 1 and type 2, there is in all of these Usher forms, there is an association with sensorineural deafness and vision loss due to retinitis pigmentosa. Patients affected by Usher syndrome type 1 and also some patients affected by Usher syndrome type 3 have in addition a third sensory defect, balance problem due to vestibular defect. And I like to remind you that both the vestibular organ, the balance organ, and the cochlea together forms the inner ear. Our study of the Usher syndrome led us to the auditory sensory cell, the air cell, and more specifically to their sensory antenna called the ear bundle. Here is an open cochlea. At the surface of the sensory epithelium, you can see the ear bundle of the air cell, and above them, the tectorial membrane. At the front, the ear bundle of the outer air cell, at the back, the air bundle of the inner air cell. The air bundle of both the inner and outer air cell performs a mechanoelectrical transduction, that is, they convert their sound-induced mechanical stimulation into electrical signal. However, inner and outer air cell have very different roles. Inner air cells are the genuine sensory cell. Their receptor potential modulates the synaptic release of the neurotransmitter to the afferent neuron. By contrast, outer air cells are sound amplifiers. Their receptor potential modulate their amplification of sound evoke stimulation. At the top left, an air bundle from a mature outer air cell. It consists of stereocilia, actin field process organized in 3 rows of increasing height and forming a staircase pattern. As shown below, during the morphogenesis of the air bundle also contains a true cilium, the kinocilium. A number of fibrous links connect the stereocilia together, but only one of these links had previously attracted attention. The tip links you see here, a single link that extend radially between the stereocilia row. As indicated here, this is indicated here by the dash red rectangle. So this link extends obliquely from the small and the middle-sized stereocilia row to the immediately adjacent stereocilia belonging to the immediately upper row of stereocilia. The key role of this link in mechanotransduction was well established. By contrast, the other links have been neglected. The other links are multiple lateral links. There are at least 6 types. Some are embryonic lateral links, here in one, which connect together the stereocilia emerging from the apical surface of the air cell. Also, during embryogenesis, there are some kinociliary links that connect together the kinocilium to the stereocilia. In addition, there are other links, which are of 4 types, which are also lateral links from 3 to 6, and which appear after birth. They run horizontally and connect together the stereocilia within a row and between rows. In 3, you can see one of these type of links, the ankle link, that connect together the base of the stereocilia, and they are only transiently present after birth. Next, you can see some links which are, let's say, present, which are called shaft connectors, and which are forming mesh-like structure. And finally, you can see here 2 other types of links, and they are very special in the sense they are only present in the outer air cell and not in the inner air cell. They are of 2 types. First, in 5, the attachment link of the air bundle to the tectorial membrane. They connect the tip of the higher stereocilia row to the tectorial membrane. And the other type of links are the top connectors, which are connecting together, and they are here indicated by white arrows, the stereocilia within rows and between rows. As I was mentioning, the role of these lateral links was entirely unknown. We first studied Usher syndrome type 1, and we identified 3 of 5 causal genes. The Usher1 gene encodes 2 unconventional cadherin. These are transmembrane protein, mediated calcium-dependent adhesion, and which have long extracellular domain composed of 27 extracellular repeats for cadherin 23, the longest form, and 11 cadherin repeats for protocadherin 15. 2 other Usher1 genes encode predicted scaffolding protein, harmonine, a PDZ domain containing protein, and SANS, an ankyrin repeat domain containing protein. And finally, the large gene encodes a motor protein, an unconventional myosin, myosin 7A. Please note, black star at the end of the C-terminus, which indicate the presence of a PDZ binding motif, also termed PBM. all the Usher1 protein except myosin 7A and the C terminal of the harmonine isoform C carry a PDZ binding motif. So first, we try to see whether or not these 5 proteins, the 5 Usher1 protein, could form a complex, and we explore at first step whether or not they co-localize in the stereocilia, and especially we could see that they co-localize in the hair bundle, in the inner and the outer hair cell, and they were co-localized in the hair bundle here during the development of the hair bundle. We then examine whether or not they lead to similar phenotype when the corresponding gene is knocked out in mouse mutant. And we observe that this mouse mutant for each of the 5 Usher1 genes have very similar abnormalities. From early stage of embryonic hair bundle development at embryonic day 17, E17, the stereocilia, unlike those of the control, were not connected. This is shown here for mouse mutant defective for cadherin 23. At early stage, the stereocilia form independent clumps. Importantly, in every Usher1 mouse mutant, one or several of the other Usher1 proteins were mislocated in the hair bundle, suggesting that they normally interact together. Cadherin 23, as shown here by immunogold labeling, form fibrous link, interconnecting the stereocilia together, and also fibrous link interconnecting the stereocilia to the kinocilium, all of these during development. Guy Richardson could show that protocadherin 15 has the same pattern of expression, from which we propose that both cadherin 23 and protocadherin 15 form embryonic link, let's say the interstereociliar links and the kinociliary links. And regarding to the 3 other Usher1 proteins, since harmonine B, we showed, is able to bind to the actin filament, same thing for myosin 7A, we propose that the 3 other Usher1 proteins, harmonine B, myosin, and SANS, form a complex that anchor this link to the actin filament of the stereocilia. And as a conclusion, we also propose that this link are essential for the early cohesion of the hair bundle. Let us move to the mature hair bundle, and let's say to the auditory transduction. Here is a diagram of 3 rows of stereocilia showing only one stereocilium per row, and the tip link. The mechanotransduction channels, here indicated as MET, are located at the base of the tip link. And upon positive deflection of the hair bundle, that is towards the tallest stereocilia row, the tip link tension increases, opening the mechanoelectrical transduction channel. This results in a robust influx of cation, mostly potassium and calcium, which trigger hair cell depolarization within a few microseconds. This depolarization led, let's say, also to the opening of the calcium channel at the base of the hair cell, resulting in an influx of calcium at the level of the synapse, which trigger the fusion of the synaptic vesicle with the plasma presynaptic membrane, and thus result in neurotransmitter release. Patch clamp recording of the mechanoelectrical transduction current here shown lead, let's say, to a mechanoelectrical transduction currents that varies with the deflection, and this establishes the relationship that you can see here between peak current and hair bundle displacement. And the sensitivity of hair cell transduction is measured from the slope of the curve. So we decided to address a possible later role of Usher1 protein in mechanoelectrical transduction. To this purpose, we had to prevent the early morphological abnormalities of the developing air bundle of Usher knockout mice. For this purpose, we developed postnatal conditional Usher1G knockout. Here, a conditional knockout for the Usher1G gene encoding the scaffolding protein SANS. The air bundle has shown here as a normal V morphology. A comparison of the ex vivo mechanoelectrical transduction current recording in this mutant mouse in blue versus the control in black showed that the sensitivity of the air bundle is preserved. Let's say also the adaptation of the currents, let's say the way the current persists, whereas let's say the stimulation is maintained or persists or not, was not different in the mouse mutant and in the control. However, we could observe a clear decrease in the amplitude of the mechanoelectrical transduction in the conditional knockout mutant for the Usher1G gene. The simplest hypothesis would be a loss of the tip link. And this was indeed what we observed by electron microscopy. One day after the loss of SANS, immunolabeling, the tip link disappeared. So we could conclude that SANS is required to maintain the tip link. And this work led to a major observation in parallel. We observed that as a result of the disappearance of the mechanoelectrical transduction, a systematic regression of the short and middle-sized stereocilia rows was observed. So this provides the first experimental evidence for a link between mechanoelectrical transduction machinery and stereocilium actin dynamics. In other words, the air bundle activity controls the maintenance of the air bundle staircase pattern. We extended our study of possible role of Usher1 protein in mechanoelectrical transduction to protocaderine 15 that forms the basal part of the tiplin to assess which isoform of the basal part is essential at mature stage. Similarly, conditional post-Laut deletion of the protocaderine 15 isoform, each of the isoform, which differ only by their intracytoplasmic region, showed that the CD2 isoform was the only isoform required at the basal part of the mature tippling at adult stage. And its loss again led to the regression of the small and middle-sized stereocilia. Elise Pepperman was responsible for this finding during her PhD in our laboratory. She is now working as a postdoc in the laboratory of Geir Bagerman in the Center of the Proteomics in Amherst University, and will later, during the day, present a result addressing the composition of the mechanoelectrical transduction machinery by a complementary approach. We also found that a few days after birth, harmonine B, the longest isoform, was present exclusively at the upper insertion of the tippling. So this suggests that harmonine B has not only a key role during the development of the air bundle, but also later on when the mechanotransduction machinery starts to work. And we could show that indeed this isoform B, which binds to the actin filament, has an essential role. Taking advantage of the conservation of the mechanoelectrical machinery in both auditory and vestibular air cell, and taking advantage that in mouse mutant only for the B isoform, the air bundle of the vestibular air cell is entirely preserved, we explore the physiology of the mechanotransduction machinery in the vestibular air cell. And to cut a long story short, we could show that the amplitude of the current is normal in the absence of harmonic B, but the adaptation process, the closure of the mechanoelectrical transduction when the stimulus is maintained was affected. And we went to conclude that the decrease of the tension exerted on the mechanoelectrical transduction was therefore greater in the absence of harmonic B, in the absence of harmonic B. So this suggests that harmonic B connects the upper part of the tip link to the actin filament of the stereocilia and thereby limit the relaxation of the tip link. We developed in parallel biochemical approach, and we explored direct in vitro interaction between the various USHR1 protein by surface plasmon resonance and identified binding partner by generating inner ear yeast to hybrid cDNA library. We observed that the cytoplasmic region of cadherin 23 here in green interact directly to harmonine. And we have previously shown that harmonine B binds to the actin filament, so this created a direct link between the upper part of the tip link composed of cadherin 23 and the actin filament via harmonine B. We could also show that harmonine directly interact with myosin 7A. This is not shown here. And finally, we could show the existence of a tripartite complex between the cytoplasmic region of cadherin 23, myosin 7A, and harmonine. So, also, we observe a direct interaction between harmonine and SANS. So this indicates a key role for harmonine. So focusing on harmonine, we found that its C-terminal PDZ binding domain, PBM, could bind to the first PDZ, its first PDZ domain, the PDZ1, thus possibly forming an intramolecular interaction that should result in a close configuration. Small angle X-ray scattering with the complete forms of harmonine shows that indeed it has a close configuration, but a truncated form of harmonine with a deletion of only the last amino acid forming this PBM has an extended configuration. And in addition, we could show that the switch between the closed and the open configuration can be obtained in presence of SANS, which as a result give access, increase, let's say, the interaction of the cytoplasmic region of cadherin 23 forming the upper part of the tip link to harmonine. So this suggests that SANS may regulate the binding of cadherin 23, the upper part of the tip link, to harmonine B. In other words, SANS can regulate the intracellular connection of the tip link to the actin filament of the stereocilia. To sum up, we have seen that the 5 USHR1 protein interact together to form two types of embryonic links, interstereocilia link, kinociliary stereocilia link, and also the tip link, as well as their anchoring to the actin filament of the stereocilia. Based on this result, we investigated whether this USHR1 protein complex could explain the retinitis pigmentosa of USHR1 affected patients. I'd like to remind you that in mouse model, defective for USHR1 gene, there is no phenotype at the level of the retina. To cut a long story short, we found that the 5 USHR1 protein are associated with calyceal processes that you can see here at the level of the rod. These are actin fill process surrounding the base of the outer segment of the photoreceptor. This process of unknown function are also virtually absent in mouse photoreceptor cell. In Xenopus tropicalis, which possess such a calyceal process, using a knockdown approach, we show that the calyceal process are indeed associated with USHR1 protein, and that they form fibrous link that control the size of the rod disc and lamella, thus providing an holistic view of the USHER1 protein complex in both the cochlea and the retina. We extended this approach to the causal gene of Usher syndrome type 2, and briefly, using USHER2 knockout, we found that USHER2 protein form another type of fibrous link, the transient early link, the uncle links, indicated here by black arrow, and we could show, first of all, that they generate almost the same type of complex as USHER1 protein, composed of transmembrane protein with a long extracellular domain, 2 PDZ domain containing protein and myosin 7A, and the knockout mice of 2 of these USHER1 proteins separately lead to the absence of this uncle link, and as a result, the opening of the mechanotransduction channel, instead of, let's say, being performed only when the air bundle deflects in the direction of the long higher stereocilia, let's say, the mechanotransduction channel can be obtained when it deflects in the direction, in the opposite direction. So in other words, directly or more likely and directly, these uncle links control the establishment of the functional polarity of the hair bundle. Our study of hair bundle links also identified the second most frequent gene implicated in deafness, which encodes a protein we call stereocylin. In the 2 panels at the top left, we can see horizontal top connectors, as I mentioned previously, which connect the stereocilia of the outer hair cell bundle between rows and within rows. We observe by immunolabeling that stereocylin is a component of the top connectors that bridge together the stereocilia within rows and between rows. Also by immunolabeling, we observe this immunolabeling at the very tip of the highest row of stereocilia, shown here, and in parallel, the imprint of these stereocilia in the tectorial membrane, which are also immunolabeled by stereocylin. So altogether, this shows that the top connectors and the attachment link of the highest row of stereocilia of the outer hair cell to the tectorial membrane are composed of stereocylin. So this summarizes this. This is a scheme for chinchilla outer hair cell showing, first of all, that the 2 types of link are conserved. Here the attachment link to the tectorial membrane and the top connector composed of stereocylin. The knockout mice for stereocylin led us, as shown on the left, to a misalignment of the very tip of the stereocilia, here in the mouse mutant for stereocylin, STRC --. At upper magnification, we can see that the top connector here indicated by black arrows are absent in the stereocylin deficient mice. But the tip link, here indicated by black arrows, persisted. And in physiological terms, we face a very uncommon situation. As shown here on the left, at the hearing onset, the hearing threshold of the stereocylin deficient mice was in the range of the normal response of the control. But we could observe a complete absence of the distortion product of photoacoustic emission. These are sound emitted by the cochlea in response to 2 tones of frequency F1 and F2, here shown in blue and red. And let's say this sound emitted by the cochlea are indicated by the green wave with the most prominent distortion product, the 2F1 minus F2. So they propagate retrograde and can be recorded by a small microphone introduced in the outer canal. So why such a phenotype? How can we explain such a phenotype? The normal hearing threshold indicated that the mechanoelectrical transduction is conserved and that amplification is also conserved. So why the absence of distortion product? What could be the interpretation of this very curious observation? When a stimulus opens one mechanotransduction channel, it triggers a coordinated opening of a cascade of mechanoelectrical transduction channels. Our results suggest that in the absence of stop connector to maintain a tight cohesion of the stereocilia within the outer air cell air bundle, the release of the mechanotransduction channel cannot, let's say, of the gating spring which operate on the mechanotransduction channel cannot properly operate. And as a result, there is an absence of nonlinear activity in the air bundle and thus the absence of generation of distortion. These results have clinical implications. The absence of distortion products which are recorded in clinics may have diverse origin and should no longer consider as exclusively due to defective amplification. As shown here, they can result from a normal coordination of the opening of the mechanotransduction channel. Following the same strategy as before, we try to pull out genes, let's say, which may belong to the same molecular complex as stereocylin that is forming the top connectors and the attachment link to the tectorial membrane. And we could come up with two forms of two deafness genes for which the mouse model indicate a similar phenotype. These genes encode autogelin and autogelin-like, and in knockout mice, for each of these genes, the corresponding gene, there is no attachment link and no top connector. So together, this finding indicates that these two types of outer air cell link compose the stereocylin, let's say, are the same component, stereocylin, autogelin, and autogelin-like, and thereby likely share mechanical properties. In summary, thanks to the genetic approach, the molecular composition of the air bundle links and their anchoring complex to the actin filament has been at least partly elucidated, and the role of this link partly also elucidated. We have developed similar approaches for elucidating, let's say, some mechanism underlying the physiological property of the air cell synapse, some aspect of the cochlear ion metabolism, the resilience of the cochlea to sound stress, and other. And unexpectedly, we found that some of the deafness genes that play a key role in the cochlea have also a critical role in the development of the auditory cortex, potentially limiting the success of cochlear therapies. As mentioned by Guy, in parallel, we decipher the pathogenic processes underlying inherited forms of deafness. And today we propose the following classification based on the same cell type from 1 to 4 and based on the function they are involved in from 5 to 7. Most forms of deafness affect the air cell, their air bundle, their synapse, the junction with their neighboring supporting cell. Deafness genes are also involved in cochlear, ionic and metabolic homeostasis, oxidative stress, inflammation and immunity, and transcriptional regulation. Armed with this knowledge, we considered ourselves ready to attempt to prevent and cure deafness. Much of what we know concerns monogenic forms of deafness. We therefore first consider to try to tackle the causes of these hereditary forms of deafness by developing gene therapy approaches. And we have so far focused on gene replacement for curing the autosomal recessive form of deafness, the DFNB form. A few years ago, we started by studying the restoration of hearing by gene replacement in mouse model for human deafness. We studied the 4 monogenic forms of deafness shown here. all are autosomal recessive forms, the DFNB form, all are hair cell defect. In all of them, we use adeno-associated virus mediating the cDNA transfer and that we injected within the cochlea via a recombinant vector. In the first three forms, corresponding to an Usher1 type 1 form, an Usher3 form, and the genetic form, the form that Guy just mentioned, leading to an hypervulnerability to sound, viral transduction in the corresponding animal models was performed at early postnatal stage before hearing onset. So this has to be extremely clear. In other words, we try not to cure, but to prevent the hearing defect. Partial prevention was achieved in the two Usher models. This partial prevention was probably due to the hair cell damage already irreversible at the time of the intervention. In the mouse model of hypervulnerability to sound, we were able to achieve the full prevention following the complete elucidation of the underlying pathogenic process mechanism. The causal gene encodes pegvakin, which we show to be involved in pexophagy, that is the autophagy of peroxisome. Pegvakin recruits the autophagy machinery, triggering the degradation of peroxisome that has been damaged by sound-induced oxidative stress. This recruitment involves the direct binding of pegvakin to LC3B, an autophagosome-associated protein. Viral transduction of the auditory hair cell with both, and only when both were associated, pegvakin and LC3B cDNA, resulted in a full prevention of noise-induced hearing loss. So this is just to show you the injection within the cochlea. Most of the gene replacement therapies in mouse model of human deafness today have involved intervention in early postnatal period, as I was mentioning, to prevent the development of hearing impairment. Here we touch on a major issue. The therapeutic window for each form of deafness must be defined. Mouse models for human deafness are generally used, but this is problematic because the maturation cinetic of the human and mouse cochlea are quite different. The mouse is born deaf, and its cochlea does not begin to function until postnatal day 12. Whereas the human fetus cochlea begin, let's say, the human fetus begins to hear in utero at 25 weeks of gestation. Crude extrapolation suggests that for this gene therapy in humans to be successful after birth, effective hearing restoration must be demonstrated in mouse model of human congenital deafness for inner ear injection performed at about postnatal day 20 onwards. So, for the first two deafness forms, the Usher form that I discussed, the timing of the gene therapy has been shown to correspond to, let's say, to a therapy that should be done in utero. And so far, there are only two deafness forms for which the reversion of the phenotype has been obtained by postnatal treatment in the mouse at least after postnatal day 20, meaning that it corresponds to a postnatal therapeutic window in humans that could be, let's say, that can take place, that can be used, let's say, after birth. These two genes, which have such a profile, are the gene encoding the glutamate transporter VGlut3 and otoferlin. We concentrated a lot of otoferlin. This is one of the first causal genes for congenital profound deafness we identified. It's encoded ferlin which is a transmembrane protein and which has a C-terminal transmembrane domain and several C2 domain binding calcium. And briefly, we could show that this protein, let's say, has a key role at the level of the synaptic exocytosis in the inner air cell. We could demonstrate that this protein is associated to the synaptic vesicle and that it controls the calcium-dependent fusion of the synaptic vesicle with the presynaptic membrane, as well as the recycling of the synaptic vesicle. By using a mutant strategy in which we were able to neutralize some calcium binding site in the C2C domain, we could, let's say, confirm the initial hypothesis and the conclusion regarding the role of otoferlin at the level of the synapse, both in the recycling of the vesicle and the synaptic exocytosis. Otoferlin cDNA has a length exceeding the 4.7 kilobase carrying capacity of the adeno-associated virus. However, the cDNA can be split into two separate adeno-associated viral vectors, both sequence ending by an inverted repeat. The two vectors carry the two separated halves of the cDNA. The 5' vector contains the promoter and the 5' half of the cDNA and the splicing donor, SD, and the 3' vector contains the splicing acceptor site, SA, the 3' half of the cDNA and the polyadenylation signal. The full length of the cDNA is reconstituted by tail-to-head, let's say, concatenation of the two recombinant viral vector, and let's say by the splicing between using the donor and the acceptor splicing site of the mRNA molecules. So doing, on a collaborative basis, we were able, let's say, to restore the expression of otoferlin in the inner air cell. And when these vectors, the two vectors, were transferred in knockout mice for otoferlin, we obtained a complete reversion of the phenotype. And this reversion could stay very long. We have now results lasting on, let's say, at least 1 year. So one of the key issues for using gene therapy for deafness is at least to have a result as effective as the cochlear implant, the only option available today for, let's say, this is a corrective or a palliative option, but not a treatment. Also, we need to achieve a long restoration that I already mentioned, and we need to have a safety procedure. And adenovirus associated injected into the cochlea, let's say, have been shown to reach the contralateral cochlea. So this is a real concern. This indicates a diffusion out of the cochlea in the cerebrospinal fluid via the cochlea, as shown here, raising the possibility of the therapeutic agent diffusing into the brain parenchyma. In conclusion, we have already obtained some promising results from future gene therapy for deafness. However, interesting challenge remain ahead to ensure safety and finely tuned restoration of cochlear function. And before closing, I wish to warmly thank my colleagues and our collaborators at the Institut Pasteur who contributed to this work on the left and our collaborator on the right, with a special thanks of Paul Avant. Also, my special thanks to the deaf family we could study who contributed to this advance. And their generous contribution has been really very decisive in the progress that we have been able to do. And many thanks to you for your attention.
Okay, Christine, thank you very much for your impressive presentation showing the meticulous work and the high complexity of the antennas of the ear, the hair bundle complex, and also your first successes into gene therapy in the mouse. We have a number of questions for you. Let me go to the list here by the chat. Yeah, there is a first one, and it says, at present there are many solutions for hearing loss, including cochlear implants, and they have shown to have very good results. And the question is, how does a potential cure, I think it's assumed here a gene therapy cure, differ from these solutions, and what could still be gained?
Okay, so what I'd like to point first is that cochlear implant, which is used for very severe to profound deafness, is not a treatment. These are prostheses. They are, let's say, they compensate, okay, the hearing defect, and they are electroacoustic devices in which bypass the external ear, the middle ear, and the cochlea, and which directly stimulate the auditory nerve. So this is basically a neuroprothesis. And let's say, so what is important in the success is whether or not the patient, let's say, have sufficient auditory nerve with a normal functionality to be stimulated in a proper way. But let's say this is completely different from what is proposed by gene therapy. Gene therapy approaches propose to cure the cells. That is, having identified that the gene is responsible, is causative for deafness, the objective is to restore the function of these genes. So this can be done by several ways. We are so far mainly focused on gene replacement therapy, in which applies specifically to the situation in which the two alleles of the genes are non-functional, or let's say, so these are recessive form, or let's say dominant form of deafness, in which the inactivation of one allele results in haploinsufficiency. In this replacement model, the objective is just to transfer the cDNA encoding. We expect the optimal situation is the situation in which the gene encodes only an isoform. And having, let's say, this cDNA encoding the normal protein being vectorized by adenovirus recombinant vector and transfer within the cochlea, and especially in the appropriate cells, and as we have seen in a number of situations in hair cells, we hope to be able to prevent, let's say, or let's say, to cure hearing impairment. So this is a completely different strategy.
Mm-hmm. Okay.
But that does not mean that if gene therapy has success, and we hope that it will be great success, especially because using, let's say, cochlear implant, as you mentioned, great success has been obtained. However, what the cochlear implant is not good is at being able to confer hearing in noisy environment. Also, listening to music is defective by cochlear implant. Listening to speech, understanding speech, is really very good through cochlear implant. But what we expect by curing really the cochlea is to restore hearing in noisy environment and also the possibility to enjoy music. And of course, people will be very pleased not to have, let's say, all this external device and even all this wonderful device being present outside and within, let's say, at the level of the temporal bone.
Yeah. Of course, yeah, thank you. There is another question, and it's actually a very simple question, but maybe also complex. So somebody asks, why is the ear so complex? And I think it's a good question. You have all these different molecules, all these genes working together. If we compare it maybe to a microphone that man made, much simpler, why is it necessary that nature made something so complex?
This is complex, okay, but let's say what the hair has to analyze is also very complex, okay? Yeah. And there are a very simple version of hearing. We think, for instance, that in non-vertebrate animals, especially in sea animals, they are, let's say, at the level of the tentacle, a mechanosensory system which is able to detect waves generated by prey or predator surrounding, for example, these sea animals. And it has exactly the same system, which is not so sophisticated in itself, meaning I want to refer to the mechanotransduction channel and the tip link. Yeah. This is very simple, in fact, but what is much more complex is the fact that each cell, not in sea animal, but in the cochlea, in vertebrates and in mammals especially, each cell differs from one to the other. Yeah. Each cell is tuned to a specific frequency. So that does not mean that this is very complex in itself, but that each cell somehow has to be tuned to a specific frequency by specific, or let's say, increasing probably some parameter stiffness, length of the stereocilia, and so on. So there is a fine-tuning of the morphological property, the physiological property of in parallel. Yeah. So, and also what is complex, and this is why we have to explore hearing and also to decipher the hearing mechanism in vivo. Yeah. That is, what you need is you need to have, let's say, the play between the various physical forces which are, let's say, induced by sound and which travel along, let's say, the cochlea, all along the cochlea, and also there are forces in various directions. So in terms of mechanical forces, this is really complex. But the basal model, I think, we try to decipher it in C anemone. We will see where it goes, but we would like to have the minimal model to, especially to understand what has been incremented thereafter to get a more sophisticated organ.
Maybe very quickly, last question. Somebody asks, why specifically you use mice to study, and is indeed the ear of the mouse so similar to human?
This is an excellent question. It's not, okay. It's similar and dissimilar. We use mice not because that it is the best animal model for studying hearing, but because we can genetically modify mouse that is still very difficult in other mammals. For instance, if we want to use gerbile, that could be possible because the genome has been sequenced. But for sure, all the technique, all the mice mutants have been developed. All the mutants have been developed in mouse, and now almost all the gene, or soon all the gene present in the mouse genome, will be, let's say, will be modified. Okay, meaning that they will be at least ready to be explored as knockout mice. So I think that this is the reference model in biology. And for sure, it will be very much exciting, let's say, to be able to develop other genetically modified models which have an hearing frequency spectrum, that match better the frequency that, let's say, mammals, let's say, especially humans can hear, and not, let's say, the high frequency that mouse can hear. Especially language relies a lot on low frequency hearing, and mice do not hear low frequency.
Yeah. Okay, maybe to finalize, a personal question, Christine. You are a very successful researcher. What would be your advice to young researchers if you would give them a 1-minute advice or 1 or 2 sentences advice? What created your success?
Try to identify where are the key questions to solve, and try to be very independent intellectually. Try to find niche also, niches, and basically try to be as much as possible free in your research approach.
Yeah, okay. Thank you very much for this beautiful presentation and all the clear answers. Thank you.
Many thanks to you. Many thanks to the audience.
It is now time for the official conferral of the honorary degree. In a few moments, nominator Professor Guy van Kamp will deliver the laudation. Afterwards, Honorary Dr. Petit gives her own word of thanks. But before all that, let's watch the introduction video together.
Wij willen graag het eredoctoraat geven aan Christine Petit voor haar belangrijke verwezenlijkingen bij het ontrafelen van de moleculen en de moleculaire mechanismen die aan de basis liggen van het gehoor. Daarvoor heeft zij families met erfelijke slechthorendheid onderzocht en daaruit de plaatsen en de genen gehaald die die slechthorendheid veroorzaken. En vervolgens is zij die genen gaan bestuderen met allerlei mechanismen en manieren en is erin geslaagd om de netwerken waarin die genen werken te ontrafelen en de werking van die genen en die moleculen op te helderen. En dat is erg belangrijk natuurlijk, ook als we denken aan mogelijke therapieën om dit op te lossen. En dat is toch een heel belangrijke verdienste. Haar onderzoek heeft al belangrijke gevolgen op dit moment. We kunnen nu al door haar onderzoek een genetische diagnose stellen bij kinderen bijvoorbeeld die doof geboren worden. Maar belangrijker nog, we kunnen nu beginnen te denken over genetische therapieën. Tot nu toe hebben we eigenlijk slechthorendheid opgelost via gehoorapparaten, cochleaire implantaten. Dat is elektronica die het geluid versterkt. Maar nu kunnen we denken aan een gerichte biologische oplossing, een gentherapie die momenteel erg in de belangstelling staat. We zullen hopelijk in de toekomst die kinderen kunnen behandelen, genezen misschien. We zullen misschien bij mensen op latere leeftijd kunnen voorkomen dat ze slechthorend worden, dat ze gehoorapparaten nodig hebben. En dat zou heel erg baanbrekend zijn. En daarvoor heeft zij eigenlijk de basiskennis aangeleverd. Er zijn zeker raakvlakken met de Universiteit Antwerpen. In mijn onderzoeksgroep doen we gelijkaardig onderzoek, vooral het genetisch aspect dan. Dus het onderzoek gaat zeker nog verder, ook samen met haar hier in Antwerpen. Zij heeft zeker de wereld veranderd en verbeterd. Zij is toch een van de grote op internationaal vlak die toonaangevend is en die tot de allerbelangrijkste ter wereld mogen gerekend worden op dat vlak. En zij is echt een voorbeeld voor mij. Ik ben heel erg blij, want het is iemand die het zeker verdient. Zij heeft heel hard gewerkt. Zij is een echte wetenschapster die een visie had, die iets heeft willen ophelderen en er alles voor gegeven heeft tijdens haar carrière en daarin ook heel succesvol geweest is. En zo iemand verdient het, vind ik. De levenskwaliteit van veel mensen, vooral kinderen, is sterk verbeterd en dat is mede dankzij Christine Petit.
Professor Petit, dear Christine, over the last 25 years, I have witnessed your efforts to understand how hearing works, from the front row. You went deeper than others, more fundamental, really trying to understand it, the traditional way, through ingenuity, originality, hard work, and personal perseverance. And it worked. The knowledge that was gained through your work already has changed many lives, and I am convinced that it will change many more through future therapies based upon this knowledge. And for these reasons, I request that the Rector honors you, Professor Christine Petit, with an honorary degree from the University of Antwerp.
Chers collègues, je vous adresse quand même quelques mots aussi en la langue de Molière. I request that the rector honors. Je voudrais quand même dire aussi que être recteur est exceptionnel. On a le privilège de pouvoir rencontrer des personnes les plus exceptionnelles dont vous. C'est pour nous vraiment un grand privilège de pouvoir vous décerner ce diplôme de docteur honoris causa. C'est ce que je voudrais quand même vous dire. On est reconnaissant pour le travail exceptionnel que vous faites et qui quand même a un impact énorme sur la santé mondiale des personnes. C'est ce que je voulais vous dire aussi personnellement. Maintenant donc la partie très officielle. On the recommendation of the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences and by decision of the Executive Board, the University of Antwerp confers upon Professor Christine Petit, professor at the Collège de France and holder of the Chair of Genetics and Cellular Physiology, head of the Department of Neuroscience, Pasteur Institut, Paris, the degree of Doctor Honoris Causa in Biomedical Sciences, in recognition of her role in elucidating key molecular mechanisms of hearing, paving the way for the development of better treatments for hearing loss. Dear Professor Avant, Paul Avant, researcher in auditory cognition and psychoacoustics at Université Clermont-Auvergne, on behalf of the University of Antwerp and the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences, I would like to thank you firstly to participate in this ceremony, and can I ask you to hand over the University of Antwerp's Honorary Medal and Diplom of Doctor Honoris Causa in Biomedical Sciences to Professor Christine Petit.
Dear colleagues, on behalf of Professor Van Houten and Professor Van Kamp, it's my honor to give this beautiful medal to Christine. Christine. Okay, and the diploma.
Many thanks.
Thank you. Thank you.
Yes, thanks.
Thank you very much. Honorary Dr. Petit, can I invite you to express some words? Thank you very much.
With pleasure. First of all, I'd like to warmly thank you for managing to create the moment of exchange in these rather difficult circumstances. I have enjoyed it very much, I must say, and I am pleased that it has been possible in some way to materialize the reception of this prestigious distinction. It is an immense honor to receive the honorary doctorate from University of Antwerp, recognized worldwide for his excellent teaching and research. The list of the recipients of this honor from your university says much about its value. Our scientific field has been marked by a profound change over the last 30 years. It has been brought into the molecular era thanks to the identification of deafness genes, which demonstrated their potential to deciphering the molecular mechanism of hearing, when we brought several disciplines together: genetics, biophysics, physiology, and medicine. This distinction shines a spotlight on the fascinating field of hearing as a key sense of human communication. The COVID-19 crisis, with the isolation created by lockdown, remind us that our species is above all a social species from which communication is vital. I homage my colleagues and our collaborators. I wish to thank them warmly, with special thanks to Professor Avant, physicist and medical doctor, who has just given me the diploma on your behalf. I have had the tremendous pleasure of working in close collaboration with him over the last 15 years or so. This honorary doctorate can also be seen as strengthening the scientific ties between your university and Institut Pasteur, particularly the Hearing Institute, of which I have the privilege of being the founding director. The first major international collaborative project in biology dates back 30 to 40 years and reveals a complete human genome sequence. Today, we expect artificial intelligence to deliver major advances in biology and medicine. But to this end, we need to work, let's say, together, and we hope to be able to work more, let's say, regularly, on a regular basis, with the University of Antwerp to accelerate the deciphering of the genetic landscape of human hearing defect within the framework of European research. Other shared challenges await us. The gene therapy for hearing loss in particular will pose ethical questions. Joint reflection will be required to reach our ultimate goal, that is, of improving the well-being of people affected by hearing impairment. Finally, I await with impatience the end of the COVID-19 health crisis so that I can meet you in person. And on a personal note, I come from a family that has lived in Burgundy for generations. Knowing about the links that existed between this region and Flanders, I look forward to learn more about your city in the near future, I do hope. Many thanks again.
Thank you very much. We wish you all the best. Have a nice day. See you later. Thank you.
I'd like to finish.
Many thanks to you.
I'd like to finish this afternoon by saying a few words of thanks. Thank you very much. Un grand merci à Dr. Christine Petit pour votre travail impressionnant, pour votre masterclass très intéressant, and of course for accepting our honorary degree. Thanks also to Professor Paul Avant, to Rector Van Goethem, to Nominator Professor Guy van Kamp, to Dean Augustijns, and to the Faculty of Pharmaceutical, Biomedical and Veterinary Sciences. Finally, of course, I'd also like to thank you all for joining us this afternoon. Let's hope that in 2022 we can once again fill the Rector Danis Lecture Hall to celebrate our honorary doctors in person. I hope to see you then.